So far this semester, what we've actually looked at are the requirements for natural selection and how that works. So we required that the traits had to vary and that those had to be inherited variations. This becomes really important because where we're going next with this material really deals with transmission genetics and the sources of genetic variation. Where do those initial variations come from? How do those get generated? A lot of this material is actually review that you should have actually learned in intro biology, but just to make sure, I want to make sure we're on the same page, and in this particular chapter, we're going to relate it to the context of evolution. So, although your textbook actually calls this transmission genetics, that really just means heredity for most of us. And the idea of studying heredity isn't new. In fact, it goes all the way back to the ancient Greek dudes like Hippocrates. He actually thought that offspring were blended from their parents. So if your father had dark hair and your mother had blonde hair, you should come out as a redhead. If your mother had a really hot temper but your father was really mild, you should come out somewhere in between. And although this idea is kind of popular, in fact, a lot of people when they look at babies say, oh, he's got his mother's eyes and his daddy's chin, this isn't exactly the way that modern heredity works, or at least not the way we understand it in a genetic sense today. So at one point in the history of humans, we actually thought that individuals contained within them individual little itty bitty teeny tiny microscopic preformed versions of all of their possible offspring. And it wasn't just all individuals, it was only the males. So the males actually contained these little tiny, what were called homunculus people. And it was when the male and the female had coitus, that's sex for the people who don't know, he transferred one of those potential offspring into the fertile garden of the woman's womb where it took root and it grew into a baby. Okay, we've learned a lot since then, and it really does start with Gregor Mendel's understanding of genetics. Even though his work was lost for a good 30 years, so about a whole generation, it really changed how we think about modern genetics and this transmission of characteristics from parent to offspring. So instead of having tiny individual minuscule people, we now actually have things like DNA, which has built on Mendel's original understanding. So in this chapter, we're going to start with a review of DNA and amino acids and proteins and how this actually helps us understand the variation and the inheritance of the characteristics of life. So that's basically, in a nutshell, what we're talking about when we talk about transmission genetics. But how does this affect the way we study evolution? Apparently, it's really important for the process of natural selection. How does mutation occur? What causes mutation? How does this make genetic variation? And how do these mutations affect the evolutionary process? Ultimately, that's really at the heart of what we need to get out of this chapter. So from the beginning, Gregor Mendel was famous for growing his pea plants. And he grew these peas from stocks that he purchased. It actually took him 3 years to develop pure breeding stocks. So he had to plant and replant his peas for 3 years before he could even begin his experiments. He actually tracked over 17 different traits of his pea plants, whether the plants were tall or short, whether the plants had flat purple flowers or white flowers, whether the seeds were wrinkly or smooth, whether they were yellow or green, whether the pods were wrinkly or smooth, whether the flowers came out along the stem or just at the end of the plant. So he looked at over 17 characteristics of the plants and he tracked them generation after generation after generation. And he kept really insanely good notes, assiduous notes. He wrote down every detail for every plant. He counted the number of peas. He recorded their color. He recorded their texture. He recorded whether that flower had been purple or white. All of these things for each individual plant in his garden over years. What's interesting is that Mendel actually wasn't a biologist. He was a math person. And he was a monk at that. And so part of this is that in the monastery where he worked, pea plants were raised in the garden and he found them interesting. And over the course of his studies, over the course of growing these plants, he thought he was maybe seeing a pattern in how these characteristics got passed on to the offspring. And that's really what triggered him to then test it. And because of the really, really detailed notes that he kept, he was able to actually establish a pattern that nobody had observed before. He still didn't know about DNA. He didn't know what was causing this pattern, but he did come up with the idea that it should have a physical thing in the plant that is causing this transmission of characteristics. So it had to be a physical thing. Today we call it DNA. And that it had to get passed on from parent to offspring pretty much intact. He didn't know what to call those. We hadn't discovered this yet. So at this point in time, he just termed them factors. Later, as we established in the 1920s, 30s, and 40s that DNA was the molecule of heredity, it became really clear that this matched up perfectly with what Mendel had observed in his pea plants. So during these studies that Mendel did, he came up with two major critical ideas. And the first was the law of segregation. And what this deals with is how the genes actually separate during reproduction for the plant. Each gene has two copies. You inherit one from your mom, one from your dad. However, they don't come with little flags that say this was mom's and this was dad's. They are just all kind of jumbled up in your cell and they're yours. So when it comes time to make your own egg or sperm cells, your own reproductive gametes, these chromosomes, as we understand them today, actually separate during meiosis, that whole prophase, metaphase, anaphase, telophase thing, and we had 2 rounds of it in meiosis. So one copy of each chromosome ends up in each separate gamete. Some genes are dominant and they'll mask or hide others which are recessive. This is the acceptable use for the term dominant for my class. If you say a trait is dominant, I assume you mean it is like big D dominant, not little d recessive. The other part of Mendel's work established his law of independent assortment, and this is related to the segregation thing. He did, again, this reinforced the idea that each gene had 2 copies, but he figured out that at least for the 17 traits in his pea plants that he was tracking, whether one particular gene ended up in a particular gamete didn't influence another gene's distribution. So if red hair ended up in one gamete, that didn't guarantee that blue eyes would end up in that same gamete. They were totally separate things to him. And this has been upheld. There are some slight variations on this. There are some slight violations on this, and we're actually going to look at that. The genes actually have to be unlinked, and I'll describe that a little bit later as we can work through this. So prior to Mendel's work, like I said, Hippocrates all the way through until Mendel in the 1800s thought that inheritance was basically a blend of parental characteristics. If that were true, what would happen is basically what you kind of see here, yellow and blue make green, right? So blending would mean that our chromosomes would actually become blended characteristics from our parents. We would end up with green traits, a yellow trait and a blue trait. It would create a whole new trait that was green in color. What Mendel's work showed is that this isn't the way it works. The yellow trait and the blue trait both remain, and it's how they actually play together that determines the final color. So this is actually a model of particulate inheritance, and this is what told Mendel that these traits were physical things. They didn't mix, they didn't change characteristics, it was just how those two things interacted with each other that determined the final outcome. So our phenotype, the physical characteristics, our skin color, our hair color, they can be intermediate. They can be a blend of characteristics, depending on the genetics. But the genotypes don't actually blend. What we end up with are things like big A and little a. And those remain separate. They do not create a new sort of hybrid A-ish, A and a half-ish characteristic. This is important from an evolutionary perspective too, because think about it. Humans have been breeding for a really long time, many thousands of years at this point. If inheritance were blended for all of this human history, at this point, we should all be clones of each other. We should all have the same skin color and eye color and hair color because all of this should have blended out by now, and we should all be pretty uniform and characteristic, which is what they're actually showing us in example A here, is what you would expect to see for blended inheritance. And that's not what we see. What we actually see is that that variation gets maintained from generation to generation, even if it's hidden for a little while, like it is in the F1 generation in this particular example. So this is important for that variation part of the picture in terms of natural selection, because it means that those genetics, those traits, are still there. They may be hidden for a little while, but these different combinations can give us new and different traits. And that variation is still there, and it's still possible for natural selection to act on it. Since Mendel's work, we've identified that DNA is the molecule of heredity. And there is actually a story behind that. Originally, when we were looking at what we thought caused inheritance, we didn't understand what it was. We had no idea. We knew about carbs, we knew about proteins, we knew about lipids, we knew about RNA, we knew about DNA, but we had no idea which of those 5 things was actually the molecule of heredity. So we had to do a bunch of studies to try to figure this out. Eventually, it turned out that it narrowed down to basically 3 main possibilities: proteins, DNA, and RNA. And it took us a while to tease that apart. Eventually, we managed to rule out DNA as being one of the molecules that, well, let me rephrase that. Eventually, we ruled out the possibility of proteins or RNA being our primary molecules of heredity. But it was confusing because those 3 things are related, and that's part of what we get into in this chapter. A small change in our DNA can have a huge impact on fitness, and we saw this when we looked at the Oldfield mice with the light-colored coats versus the dark-colored coats against the owls, and that was due to a change in one gene, that one MC1R gene that caused that coat color change from dark to light. In terms of modern human diseases, influenza is also a really good example. A single change in its DNA makes it way more dangerous to the host and much more likely to become a pandemic flu strain like the 1918 flu. So in terms of understanding what DNA actually is, you've seen this stuff before, so this should be review. Your basic DNA structure involves a 5-carbon sugar. So there's our 5-carbon sugar. It can be either ribose or deoxyribose, depending on whether you're talking about RNA or DNA, respectively. It should have a phosphate group stuck on it, and it should have some sort of nitrogen base. So this is our basic nucleotide structure up here. The bottom part are different options for those nitrogenous bases, and you'll notice that there's lots and lots of nitrogens, which is why they're called nitrogenous bases. And we have 2 different types, the purines and the pyrimidines. The purines have this double ring structure with the hexagon and the pentagon sticking off the side of it. And these include adenine and guanine. It may be kind of tough to keep those straight. I could always remember it because guanine reminded me of guano, which is bat poop, and I didn't like the structures because they were harder to draw, so bat poop stunk, purine stunk. That's just my mental equivalence for you. Maybe it'll help. But I want you to notice what's different about these 2 different bases. They've got very similar chemical structures, but look at what actually happens here and here. At the tops of these molecules, one of them has NH2, the other has a double bond to an oxygen. The only other position that's actually different is what happens there versus what happens there. This is actually what makes our DNA bases different. I mean, this is the A and G. This is half of our DNA. And it comes down to really simple chemistry groups. Who knew? When we look at the pyrimidines, these were much cooler because, well, pyrimidines, pyramids, I like ancient Egypt stuff, so that made this easier for me to remember. They were also easier to draw because they've just got the single ring structure. In this case, what I want you to pay attention to is, again, what's going on at the top of these molecules. On cytosine, you've got the NH2. On thymine, you've got the oxygen. Ooh, that's a slight change. On thymine, you've got one other thing that's different, and that's this methyl group, the CH3 group sticking off to the side. So just really tiny chemical differences is what sets up our DNA. So if you think about it, our gene sequence is based on NH2s versus oxygens and methyl groups and stuff. The chemistry actually does matter. And these sequences are what make us human. This is what makes us tick. This is what enables us to grow keratin for our hair and collagen for our skin and our eyeballs and brain cells and all of these things. So when we put together these structures, what we end up with is an alternating sugar-phosphate backbone. So you've got a phosphate, sugar, phosphate, sugar, phosphate, sugar. And this forms one, basically, the outer rung, the outer supports of the ladder, so to speak. The nitrogen bases project out into the middle of the molecule and they form the rungs of the ladder. Eventually, these twist up on each other and form the well-known double helix. What's also important to recognize on this is that the adenine and thymine base pairs form 2 hydrogen bonds, and the guanine and cytosine base pairs are a little bit stronger. They form 3 hydrogen bonds. So this is really what sets up our genetics, is the pattern of AG and TC. That's it. And it all comes down to chemistry. Now, there is one important thing that you guys all need to know how to do as biology majors, and that's actually pronounce and preferably spell deoxyribonucleic acid. And I know you can do this. You know how I know that? Because I knew a 4-year-old who knew what DNA was. His favorite party trick was to go up to adults, and his eyes would get really big because he knew he was about to do something really, really spectacular that would surprise the adults. And he'd walk up to them, and he'd go, "Deoxyribonucleic acid." He was 4. And it wasn't just that he could pronounce that. He actually could point to pictures of it in a book. He could identify it as DNA. He knew that this was the basis for our genes. He wasn't quite clear on the difference between denim genes and heredity genes, but you know, give him a break, he's 4. So he was doing pretty good. How did a 4-year-old know this? Was he some sort of super genius? No, not exactly. I mean, he's a smart kid, but his mom was a broke college student. She could afford one of 2 types of books. Children's storybooks or textbooks. Guess which one's better at putting kids to sleep? The textbook, just like it is for most people. So I know you should know this by now. You should have mastered these concepts. So we're not going to spend a whole lot of time actually looking at DNA structure. I expect you to already know that part.
In this animation, we'll see the remarkable way our DNA is tightly packed up to fit into the nucleus of every cell. The process starts with assembly of a nucleosome, which is formed when 8 separate histone protein subunits attach to the DNA molecule. The combined tight loop of DNA and protein is the nucleosome. Multiple nucleosomes are coiled together, and these then stack on top of each other. The end result is a fiber of packed nucleosomes known as chromatin. This fiber, which at this point is condensed to a thickness of 30 nanometers, is then looped and further packaged using other proteins which are not shown here. This remarkable multiple folding allows 6 feet of DNA to fit into the nucleus of each cell in our body, an object so small that 10,000 nuclei could fit on the tip of a needle. The end result is that the DNA is tightly packed into the familiar structures we can see through a microscope, chromosomes. It is important to realize that chromosomes are not always present. They form only when cells are dividing. At other times, as we can see here at the end of cell division, our DNA becomes less highly organized.
Overall, there is a flow of information in terms of how our genetics work, and that basic flow is DNA makes RNA makes protein. This is how the information that's coded for by the ATGCs get used to make actual functional working proteins that are the collagen in our skin, the keratin in our hair, our fingernails, our brain cells, all of these things. This is how that information gets passed from inside our nucleus of our cell out to the outer parts of the cell and becomes something that can actually do some work, be something useful. So structurally, DNA and RNA are a little bit different, and we do need to look at and understand the differences between the DNA and RNA. Again, this should be review. DNA is double-stranded. It uses deoxyribose sugar. Now, when I say deoxy, what does that usually mean? Oxy is oxygen. D means less or removed. So you'd expect this to be ribose with less oxygen. So look at what happens here. This is on the 2' carbon, carbon number 2 out of the sequence. They're numbered 1, 2, 3, 4, 5. So on deoxyribose, you just have a hydrogen. On ribose, you have an OH. The deoxyribose has one less oxygen, hence its name. This actually becomes important too because ribose is more reactive. Most molecules don't want to have two OH groups that close together. It makes them a little bit less stable because the oxygens are kind of negative and they repel each other. So ribose is less stable as a sugar. And this is one of the things that sets your DNA and RNA apart. The other is that DNA uses thymine while RNA uses uracil. So again, there are slight chemical differences between these two things. For example, on thymine, you have this methyl group, and on uracil, it's a hydrogen. That's the only difference between those two pyrimidines, is a CH3 versus an H. If you had to guess, which one do you think came first? I'm not going to tell you the answer to that, but I want you to think about it for a minute. So transcription is the first stage in this process of getting information from our DNA to the RNA level. And how this works is an enzyme complex, RNA polymerase, and a whole bunch of other proteins associated with it bind to a region of the DNA called the promoter. And what happens is it untwists a little part of that double helix and it starts making RNA. Your DNA actually has a direction, and the direction is really, really important because it keeps you from making mistakes. So everything that we do is going to work 5' to 3'. 5' is the head end of the molecule, 5' fist. 3' is the other end of the molecule, the tail end, elbow, part of my arm. So everything works 5' to 3', head to tail. That's going to be really important. After you make the messenger RNA, you actually have to go back and edit it, especially in eukaryotic systems, eukaryotic cell types. We have to get rid of introns. These are regions that don't directly code for proteins, and you'll learn a little bit more about those in cell biology. Not all RNA is used to make proteins. There are other types like ribosomal RNA and transfer RNA, and we'll get to those too. So this is an illustration showing you basically how this works. So in this particular top one, what we're looking at is where the DNA double helix actually unzips a little bit, it unwinds, it untwists, and this opens up a little bubble. The enzyme goes in and it starts making the RNA, it starts making the RNA in the RNA's 5' to 3' direction, so going that way in this case. This is where that becomes important because nucleic acids come together head to tail. They run anti-parallel to each other. So the 5' end, the fist end, the head end of one strand will match up with the elbow tail end, the 3' end of the other strand, and they come together in opposite directions. You guys may actually find yourselves doing this on the test. That's okay. It works. How the RNA gets synthesized, it plays match the DNA base pairs just like in DNA. So adenine bonds with uracil and RNA, don't mess that up. Thymine bonds with adenine, cytosine bonds with guanine, cytosine with guanine, thymine with adenine, adenine with uracil, thymine with adenine. And basically the enzyme just keeps adding on letter after letter after letter. It keeps adding on nucleotide after nucleotide after nucleotide until it reaches the end of the gene.
The central dogma of molecular biology, DNA makes RNA makes protein. Here the process begins. Transcription factors assemble at a specific promoter region along the DNA. The length of DNA following the promoter is a gene, and it contains the recipe for a protein. A mediator protein complex arrives carrying the enzyme RNA polymerase. It maneuvers the RNA polymerase into place, inserting it with the help of other factors between the strands of the DNA double helix. The assembled collection of all these factors is referred to as the transcription initiation complex, and now it is ready to be activated. The initiation complex requires contact with activator proteins, which bind to specific sequences of DNA known as enhancer regions. These regions may be thousands of base pairs distant from the start of the gene. Contact between the activator proteins and the initiation complex releases the copying mechanism. The RNA polymerase unzips a small portion of the DNA helix, exposing the bases on each strand. Only one of the strands is copied. It acts as a template for the synthesis of an RNA molecule, which is assembled one subunit at a time by matching the DNA letter code on the template strand. The subunits can be seen here entering the enzyme through its intake hole, and they are joined together to form the long messenger RNA chain snaking out of the top. Once a gene has been located and transcribed into mRNA, it must first be edited before it can be translated into a protein. This editing process is called splicing. It involves removing non-coding regions called introns, leaving only the protein coding exons. In the cell, the introns are removed by special enzymes which recognize specific sequences. These enzymes cut and rejoin the coding regions for translation into protein.
In the previous two slides, what you saw were a couple videos that actually illustrate the process of transcription, or how the information in the DNA gets copied into the messenger RNA, and then a really, really short one, the 30-second one on splicing, how that gets looped out and those introns get removed, so that what you're left with is a continuous piece that should correspond to the protein that you need to make. That's where the process of translation begins, and it uses all three types of RNA all at once. So let's identify where those are in this particular diagram. Messenger RNA, this is what you just made. So in this particular diagram, it's the thing that's snaking through the ribosome. You'll notice it's also got a 5' and 3' direction, and those numbers are actually noted. So the direction also matters. It gets read 5' to 3'. Just do everything 5' to 3' and it will work out. Okay. You also have another type of RNA, the transfer RNAs, and these actually contain the anticodon. It's a set of 3 bases that will actually pair up with a codon, a sequence of 3 bases in the messenger RNA. If there's a match, what happens then is the amino acid actually gets added onto the protein. So how this works out is the ribosome binds to a particular region on the messenger RNA that says start here, basically. It finishes assembling, so makes a sandwich around the messenger RNA. And then it reads the messenger RNA in groups of 3 letters. Those are called codons. So there is a group of 3 letters, GUC. That is a codon. Those codons need to match up with the anticodons in the transfer RNAs. If there's a good match, what happens is this first site you test for the codon and anticodon to match. If they do match, the whole ribosome shifts over so that this transfer RNA and the messenger RNA move into that middle position. And what happens next is you get a bond that forms between the 2 amino acids and your protein chain grows. So every time you add on another amino acid, it's like those little pop bead things that you might have played with as a kid. You just kind of stick them together and you add one onto the chain and it gets longer and longer and longer. That's how your proteins get made. Again, in the messenger RNA's 5' to 3' direction. So this will continue. It will keep going in groups of 3 letters until you either run out of messenger RNA or until you hit a stop codon. And I'll explain those too. So this is basically the master key for how translation works. This tells you what sets of 3 letters, which codons code for which amino acids. How you read this table is you start on the left-hand side and you identify your first base. So let's say my codon is ACG. I just made that up. So you look on the left-hand side for adenine. There we go. Next, you read across the top, your second base, AC. So that puts me somewhere in this block. Third base is over on the right-hand side. G is at the bottom, so ACG gives me threonine. Okay, well, that's nifty. Frankly, if you noticed, AC anything gave me threonine. So it doesn't even matter what that third base was. That's not true for all of them. Sometimes it does make a difference. But for many of these, you'll notice that there's actually duplication. Okay, one codon that you need to absolutely, positively, 100% know is AUG. That's the start codon. It codes for methionine. You need to know this one, and you need to remember this one because it's not always labeled in all versions of all the codon tables. There are 3 stop codons, UAA, UAG, and UGA. They're called stop codons because there's actually no transfer RNA that matches that. So what you end up with is nothing. It's a hole. It's a blank spot in your protein chain. You can't form a bond with nothing, so that signals the end of your protein. And that's where it stops, hence the term stop codon. You don't need to memorize those because they're usually noted in codon tables. They kind of have to be. So they're fairly easy to identify. Let's see. Another amino acid that's kind of an interesting one is UGG, which codes for tryptophan. Yes, that's Uggs, like the ugly boots, the footwear. Uggs. Okay, you may not think they're ugly. Tryptophan is an interesting amino acid, and there's a lot of biology involved with it. For years, people thought that tryptophan was the cause of getting really sleepy after eating a lot of turkey at Thanksgiving. In fact, there was even an ad on television that said, "Don't eat turkey at Thanksgiving, it makes you drowsy." As it turns out, all meat has tryptophan. In fact, tryptophan is present in a lot of proteins, so it's not actually the fault of the tryptophan. There may be some limited truth to that. Mythbusters actually tested this, and what they found was that when they gave the Mythbusters a bunch of pure tryptophan, it did make them drowsy. And it's actually included in several over-the-counter herbal type sleep aids. It doesn't work for everybody. It works for some people and not for others. So then the Mythbusters went on and they actually looked at Thanksgiving dinners, and they tested it, same calorie levels with and without turkey, and they got equally drowsy. So it kind of proved that it wasn't the turkey that was at fault. It was actually the quantity of food that they ate. When they actually reduced the Thanksgiving dinner to a reasonable portion size, they weren't as drowsy. So if you need to prove to yourself that tryptophan is not the culprit, I recommend the IHOP all-you-can-eat pancake breakfast, which is pure carbohydrate, and you'll get just as food coma tired on that as you would on Thanksgiving turkey. So this is your codon table. I will provide a copy of this on the exam. Feel free to use the ones in your book for your homework and stuff. I would caution you about using ones from the internet, and that's because I've run across several that were incorrect. The messenger RNA codon table on Wikipedia is correct, at least it is at the moment. The version that your textbook actually provides is actually round, so calling it a table is maybe a bit of a misnomer, but it works slightly differently. It still has the same information, but in this particular case, you read from the inside out. So previously we had ACG, correct? That was our codon. So that means we start A, AC, ACG gives me threonine. you still get the same results. This one often is used because it takes up a little bit less space on a page. You may run across either or both versions. It doesn't really matter because, again, you end up in the same position. Just make sure that you're reading your messenger RNA from its 5' end to its 3' end. You need to label your ends. That's really important. If you do not label the ends, you'll just work left to right, and that may not be the correct direction. So always label the 5' and 3' ends. I still do it, and I'm a PhD, but it keeps me from making stupid mistakes.
The job of this mRNA is to carry the gene's message from the DNA out of the nucleus to a ribosome for production of the particular protein that this gene codes for. There can be several million ribosomes in a typical eukaryotic cell. These complex catalytic machines use the mRNA copy of the genetic information to assemble amino acid building blocks into the three-dimensional proteins that are essential for life. Let's see how it works. The ribosome is composed of one large and one small subunit that assemble around the messenger RNA, which then passes through the ribosome like a computer tape. The amino acid building blocks, that's the small glowing red molecules, are carried into the ribosome attached to specific transfer RNAs. That's the larger green molecules, also referred to as tRNA. The small subunit of the ribosome positions the mRNA so that it can be read in groups of 3 letters known as a codon. Each codon on the mRNA matches a corresponding anticodon on the base of a transfer RNA molecule. The larger subunit of the ribosome removes each amino acid and joins it onto the growing protein chain. As the mRNA is ratcheted through the ribosome, the mRNA sequence is translated into an amino acid sequence. There are 3 locations inside the ribosome, designated the A site, the P site, and the E site. The addition of each amino acid is a 3-step cycle. First, the tRNA enters the ribosome at the A site and is tested for a codon-anticodon match with the mRNA. Next, provided there is a correct match, the tRNA is shifted to the P site, and the amino acid it carries is added to the end of the amino acid chain. The mRNA is also ratcheted on 3 nucleotides or 1 codon. Thirdly, the spent tRNA is moved to the E site and then ejected from the ribosome to be recycled. As the protein synthesis proceeds, the finished chain emerges from the ribosome. It folds up into a precise shape determined by the exact order of amino acids. Thus, the central dogma explains how the 4-letter DNA code is, quite literally, turned into flesh and blood.
So, what we've actually worked through at this point in the chapter is we've looked at the structure of DNA, we've looked at how the DNA is made into an early messenger RNA, a very brief review of splicing and how that gets turned into a full mature messenger RNA, and then how that information gets translated at the ribosome into an amino acid sequence called a protein. This is how we get from DNA to functional us stuff, or functional plant stuff, or bacterial stuff. It all works the same way. No matter what kind of organism you are, this process, this hierarchy of information remains the same. DNA makes RNA, makes protein. That part you really need to know for all of your biology classes. So while we're talking about genetics and inheritance and DNA, this brings us to good old Mendelian genetics and some fun with terminology. Yay, I know you love vocabulary words, with sarcasm. So these are terms that I need you to have a reasonably good handle on. An allele is a variant of a gene. So I have an allele for red hair. There are lots of different versions of hair color. Those are called alleles for hair color. The gene itself is the stretch of DNA that says, make some hair pigment. Your spelling of that gene is what determines the color of your hair. You can be homozygous, which means you have two copies of the same allele, big A, or little a, or you can be heterozygous and have copies of two different alleles, big A, little a. So how this works out in genetic terms, according to basic Mendelian genetics, what we ended up with was big R gives you one color, big R gives you the same color, and little r may give you a different color. This is true if your genes are simple, dominant, and recessive. What this means is that little r, when it's recessive, gets masked or hidden by the big R, the dominant trait, that other version of the allele. Usually, in most cases, dark pigment is dominant and light pigment is recessive. That's true for hair color, fur color, skin color, etc. Most of the time, that's the case. Mendel didn't look at some of the more complex situations that can come about. So I did mention that phenotypes could be blended, and this is an example of how that happens. Sometimes when a plant produces even amounts of both red pigments and white pigments, for example, you end up with a pink flower. And this would be a uniform pink color, not little splotches or dots of red and little splotches and dots of white. When this occurs and you actually get blended inheritance like that, we call that incomplete dominance. That means that big R, red, is not truly dominant over little r, recessive, white. They actually blend together. There are quite a number of traits that actually work this way. In fact, a lot of our skin color isn't truly Mendelian, light and dark. It's not quite that simple. There's a lot of examples of incomplete dominance in plants, in animals, in humans, in insects, so it's not that rare. It just happens that none of Mendel's pea plant traits that he was looking at actually fit this description. So, he didn't find it. That's okay. Punnett squares were actually invented by Reginald Punnett, not Gregor Mendel, although they're kind of inextricably linked in our minds with Mendelian genetics because that's how we use them. Now, important things about doing your Punnett squares. When you're working with these, it's important that you use the same letter for a particular trait. For example, big T in this case represents a tall plant, little t represents a short plant. The capital letter is the dominant characteristic, the recessive letter, or the small letter is the recessive characteristic. You do not use two different letters. You do not abbreviate it TS for tall, short. This does not work for me. If you do that, I'm going to think you're talking about two different genes, one for tall and one for S. What's an S characteristic for a plant? I don't know. Sepals. I'll use that as an example. So make sure you use the same letter and be consistent. It's not too difficult to do. You can have incomplete dominance or codominance, and sometimes you see different designations for those, like T1 and T2. Sometimes those are subscripts written below the T's, sometimes they're superscripts written above the T's, so you might have T1 or T1. I've seen both notations. I don't care which one you use. The reason for this is to tell us up front this doesn't follow typical Mendelian inheritance patterns. It's either incomplete dominance where you get a blend of characteristics, or it's codominant where both characteristics are expressed. So what that means in terms of our pink flowers from before is that you would actually have splotches of red and splotches of white on the same flower, but the overall effect would look pink until you looked closely at the petals. That would be codominance. Those mess up our Mendelian rules of inheritance a little bit, but the genetics still work the same. So in the next thing, I'm going to have a question for you about solving a Punnett square for a problem. Okay, back to the evolution part of this. In Chapter 3, we talked about the fact that mutation was an important source of our variation. So this is where we get to look at what mutations are, what causes them, how we generate other forms of variation. At a more macro level, there are some other processes that are involved in generating variation. We'll look at those in another chapter. So for now, we're just going to focus on the DNA level, the inherited variations that we can see. Conversion mutations are ones that may change the amino acid. So what this is taking us through is what happens when you have different kinds of mutations. So in our first example over here on the left-hand side, we have an original DNA sequence, and the DNA is TCA on the top, AGT on the bottom. You'll notice it follows normal base pairing rules. The messenger RNA transcript from that is UCA. So that means that this bottom piece was your coding strand. That's the one that you actually use to make the messenger RNA off of. Adenine bonds with uracil, guanine bonds with cytosine, thymine bonds with adenine. What UCA actually codes for, if you flip back and look at a codon table, is serine. So the amino acid serine is what gets coded for in this particular spot on the protein. Now, what happens if you actually change that last letter in the sequence, and instead of an A, you end up with a G? Well, now your DNA sequence reads AGC instead of AGT. This causes a corresponding change in your messenger RNA. Now you have UCG instead of UCA. But, wow, look at that. You still end up with serine. Remember how I pointed out in the codon table that a lot of those, it was like AC something still gave you the same amino acid? Our genetic code is redundant, and that's what that means, is that there are many different ways to get to the same amino acid. In fact, this third position in our messenger RNA strand is often called the wobble position, and mutations, changes in these letters usually have the least amount of impact on our proteins. However, there are other possibilities. So what happens if we change the middle letter, for example? Instead of AGT, we now have AAT. That gives us a messenger RNA of UUA, which actually codes for a totally different amino acid, leucine. Oops, that's a change. This is called a non-synonymous or replacement mutation, where one amino acid gets swapped out for another. Again, hopefully you've seen these before. The last kind of mutation that you can have is a nonsense mutation, and in this particular case, your AGT mutates and instead you end up with ATT, which gives you the messenger RNA sequence UAA. Unfortunately, that codes for a stop. That's one of your 3 stop codons. So your protein doesn't get anything at all added. It just truncates. It just quits at that point. This is called a nonsense mutation whenever you get a premature stop codon that is because of a mutation. I want to point out one more thing while we're on this slide. Like I said, the third position is the one that's usually the least important. As it turns out, this middle position is actually the one that is usually the most important. Mutations in that middle spot almost always cause a change in the amino acid or a stop. So those are more dangerous in that sense. You can also have insertions and deletions. These can be either in frame, which means they occur as a whole triplet and they don't mess up the reading sequence of your DNA as badly. So in example A, you've got TCA, CTG, AAG, CTA. In the bottom, you've got TCA, CTG, CTA, ATA. The only thing that's missing would be that one amino acid, and because it occurs in a group of 3 letters, it means you delete a whole amino acid and nothing else really gets messed up. That might actually have very limited impact. It might be totally harmless. This is different than what happens in a frame shift mutation, which takes a number of DNA bases that is not a multiple of 3. It could be 1 base, 2 bases, 5 bases, and that's actually what they're showing here, is they actually are showing a deletion of all 5 of those letters. And you'll notice that now our sequence of triplets is TCA, that's the same, CTC instead of CTG, maybe the same and maybe not. TAA instead of CTA, very likely to be different. TAG instead of ATA, very, very likely to be different. GC something. We don't even have a third letter at this point. Insertions and deletions of non-codon numbers, not threes, are the most dangerous because they mess up the entire sequence of amino acids that you're going to get from that point forward. And usually this is very, very destructive to the protein that you're trying to build, because basically you went from here's the recipe to gobbledygook. And that's what you get, is a protein that is functionally, structurally, nothing at all like what you should have made. You just made garbage as far as your cell is concerned. You can scale this up. You can have mutations at even bigger levels, like large-scale chromosome changes. So extra copies of all of your chromosomes, that's called polyploidy. We're usually diploid. That means we've got two sets of our chromosomes, one from mom, one from dad. But strawberries, for example, they actually have 56 chromosomes. We only have, how many? 46. And that's 23 pairs. As it turns out, though, strawberries are actually octoploid. They're 8N. They have 8 sets of 7 chromosomes. That's kind of weird. This is actually common in plants, especially plants that we've grown as crops. So that's what this bar graph is actually showing us. Look at the domesticated plants versus the non-domesticated plants and how often they actually turn out to be polyploid. So, for example, you've got in the Ariaceae, the domesticated plants have a higher bar for polyploid. For the next group, polyploid. The Poaceae, it's reverse, so they're a little unusual. The Liliaceae, domesticated, is more likely to be polyploid. The Brassicaceae, things like broccoli, more likely to be polyploid for domesticated. And the Fabaceae, not as much. So 2/3 of the plant groups that we just looked at were polyploid when they were domesticated. This is actually thought to be because of our artificial selection, because of our inbreeding and our constant replanting and regrowing from that same genetic stock. Eventually they seem to accumulate extra copies of chromosomes. For whatever reason, this causes problems in plants, or not as many as it does in humans, for example, but it can still cause some. But it's also associated with producing bigger fruits, bigger seeds, sometimes even more robust plants, which is also really kind of weird because in most animals, it's the reverse. They end up being very genetically abnormal. So looking at the level of the single chromosome and what can happen to that, there are a number of additional mutations that can occur at the level of the whole piece of DNA. Not just that one gene, but all of the genes on that same strand. So you can have a duplication event, which is where one or a group of genes end up being mistakenly copied. So instead of EF, you now have 2 copies of EF back to back. An inversion, which takes a normal sequence like DEF and flips it around in the chromosome. It kind of gets reversed and glued back in backwards. That's called an inversion. A deletion, where a whole segment of a gene or genes plural can just be lost in a chromosome. And a translocation, which occurs when a piece of one chromosome actually kind of gets mistakenly transferred over into another chromosome. So the QR genes in this chromosome basically get transferred up into the other chromosome, and that's how you end up with A, B, C, D, E, Q, R, F, G in this particular case. The question really becomes, how do these happen? What causes like a duplication or an inversion or a translocation? What's the physical mechanism for that? And a lot of this occurs during crossing over, which typically happens during prophase 1 to metaphase 1 during meiosis. And that's because this is when all the chromosomes of your cell have to line up in the middle. And as they do that, they get tangled up. and the arms of the chromosomes sometimes get crossed over each other. As they separate, sometimes they don't separate quite cleanly, and you end up with these chromosomes with bits and pieces that are broken off and that reseal back onto the wrong chromosome. So you end up with new combinations of genes. Originally, we started off with, on this first chromosome, little a, little b, little c. After meiosis, we end up with little a, little b, little c, little a, little b, big C, big A, big B, little c, and big A, big B, big C. This is a normal process that is actually an important contributor to the variation that we get. Previously, those combinations of genes didn't exist. So maybe you only had brown hair and brown eyes and blonde hair and blue eyes, and what these would actually give you is somebody with blonde hair and brown eyes, or brown hair and blue eyes. New combinations of traits can occur because of this crossing over, which is also called recombination. For some reason, this caused me initially a lot of problems in genetics. I didn't get it the first time through. I don't think my instructor explained it well, but these illustrations really help. So this actually does kind of go back to Mendel's laws, because these chromosomes are the physical basis for inheritance. What Mendel said was that we had the law of independent assortment. Each one of these chromosomes ends up going a separate way into each of 4 separate gametes. So you created 4 sperm cells, each with one of those copies of the chromosome. The second part of Mendel's laws states that these should be independent. So what happens to gene A should not affect what happens to gene C. This, as it turns out, is only partially true. So A and B in this case, we never actually generated a little a, big B. And that's because these two genes are physically really close together. So I'm going to borrow a cable for a second. Charging cable off my phone. Let's pretend this is a chromosome. If my genes are physically really close together on the same chromosome, can I tie a knot in this? Nope. Nope, that's not going to happen. Not without breaking my cable. So crossing over between these two genes is actually pretty uncommon just because they're physically so close, and it has to do with like physical being able to twist and wrap the chromosomes around each other. What if my genes are this far apart? Can I tie a knot between those two genes? Piece of cake. A little harder without picking up my fingers, but that's okay. So these two genes are much more likely to undergo crossing over. And this is something that we observed and that we've confirmed, is that the closer the genes are, the more likely they are to be kind of stuck together. This will become important later in chapter 9, so we will come back to this idea too. But this little part of that, this A and B set, since they're physically so close on the chromosome, they actually do violate Mendel's laws of independent assortment, because you don't get new combinations of those very often. However, the distance between A and C or B and C was far enough that you do get crossover between those chromosomes. So you can get genetic exchange between those chromosomes and create new combinations. Okay, so what does this have to do with fitness? That's a good question. Mutations are random. We can't control when a mutation is going to happen or where a mutation is going to happen, with a very few exceptions. Some mutations are good, some are bad, some are neutral. Which ones you get is completely undirected. So this illustration that you see here with the polar bear, it's shivering, it's cold, it's a naked polar bear. It's saying, what I need is a good fur coat. It needs a mutation for a fur coat. Is it going to get one? No, it's a dead polar bear. It's okay, it's just an illustration. We don't actually get to make mutations in response to a need. I can't mutate because I need to fly. I can't mutate because I need to become plague resistant. I can't mutate because I need to be able to swim faster. I can't control any of this. This is occurring inside my cells at a microscopic level. So it's random. And we actually need to establish that this is true. Remember, I told you in the beginning of the semester, don't take my word for it. I have to show you evidence. So researchers actually tested this using some bacteria and a phage, which is a virus that infects bacteria. And they had two different ideas for how this could work. In the first, the idea was random mutation. So if mutations are random, this is what they expected. There should already be a few virus-resistant bacteria in the population, just because of random chance. That's the random part of this. Their descendants, when they're exposed to the virus, should survive and reproduce if they've got virus resistance genes. The others should die. It's E. coli, so nobody cares if they die. The second idea was that if mutation is not random, if it's an acquired response, then initially we should observe no virus-resistant E. coli until they encounter the virus. And that after they encounter the virus, then we should start seeing these mutants, these virus-resistant strains of E. coli appear. So how does this play out? The Luria-Delbruck experiment is exactly what they did. So they actually took some virus-sensitive E. coli, they grew them up in culture, and what they did was then they spread the E. coli out onto petri dishes that had been pre-coated with layers of virus. So you had this virus-laden petri dish, and you put the bacteria on it. Then it's actually really easy because all you have to do is count the numbers of colonies that you see. That's a survivor, so it was resistant. That's a survivor, so it was resistant. That was a survivor, so it was resistant. And you can figure out how many resistant strains or mutations you actually had. So how does this help us test the hypothesis? Good question too. According to the random mutation hypothesis, what we should see is some plates that have a lot of survivors and some plates that have few or no survivors, and it should be random. You might have 30 on one plate, 300 on another plate, 0 on yet another plate. It should be all over the place. Completely just scattered data, random data. If their second hypothesis is correct and the mutations come about because of exposure to the virus, then what we would expect to see is that all the plates of bacteria should respond similarly to the same exposure to the same threat, and so on each plate we should see similar numbers of mutants. When they actually did this experiment, they saw a huge amount of variation in the number of survivors. So that actually supported the idea that mutation was random. There's your piece of evidence. This is actually one that you could do with, some really basic equipment if you wanted to. Okay, still on the topic of mutation. How often do these occur? How fast do these occur? How much of a threat is mutation to our existence? First question for you, why does DNA mutate? And I know you can't answer me because I'm sitting here talking to a camera, but these are some of the most common causes of mutations. Carcinogens, things that cause cancer. So start listing some carcinogens. Go. Out loud to yourself right now. Talk yourself through it or jot notes down if you can't talk out loud. What causes cancer? Should I hum the Jeopardy theme song? Okay, radiation is another cause of mutation, and in fact we've actually looked at the mutation rate due to ultraviolet radiation like sunburns, and we found that this causes 1 million mutations per day. That's a lot. And cellular stress, oxidative stress, it's oxidative damage. Another source of mutations is just errors in copying your DNA. You make a mistake and you get a G instead of a T. It happens. So how often does this occur? It varies. And I hate to say that because it sounds like a cop-out. It sounds like an excuse. But the simple reality is that it's different for different organisms and for different genes. It's not consistent at all throughout our genome. Part of that is because some of our genes are really, really critical. Like, here's the genes to do glycolysis. Your cells have to be able to do that. Mutations in those genes tend to be deadly. Why don't mutations get fixed? I mean, isn't there supposed to be some sort of cleanup mechanism? Yes. The enzyme that copies our DNA typically makes about one mistake out of every 100,000 bases. So what that works out to for human cells is approximately 120,000 mutations or mistakes every time a cell divides. Oops, I just killed some skin cells. They're going to have to replicate. They have to duplicate their DNA. I just caused 120,000 mutations per cell that has to regrow. Ooh, I'm going to get skin cancer. Or am I? So we have proofreading enzymes, and this is useful because they clean up 99% of those errors. So now we're down to 1 in 1,000 DNA letters that get permanently changed every time you have to copy your DNA. That's about 1,200 per cell division. I still don't like this number. This is a scary number. Then there's another tier of cleanup enzymes. Mismatch repair mechanisms come in and look for when you've got like thymine bonded with guanine, which is not supposed to happen. It's supposed to be an AT pair. So this is what this part looks for, are mistakes in base pairing and matching up. And it cleans up another 99% of those errors. So these are really good repair mechanisms. So now what we're left with is less than 1 mutation per original 1,000, or 1 mutation per cell division. So where does that mutation occur? Okay, maybe that mutation occurred in my toenail growing genes. It's not a toenail. I don't care. Or maybe it was a mutation in a heart gene. It's in my arm. Again, who cares? So where these mutations occur and how much they affect the protein sequence, that is a big part of the question. So I asked you at the beginning of this slide to list some things that cause cancer. Theoretically, you would have found it very difficult to come up with anything that doesn't cause cancer. The answers I get most often are air. Air has pollution in it. It can actually cause cancer. There are carcinogens floating around in our atmosphere. Water! Tap water may also be contaminated, so it can have carcinogens. Basically, everything can cause cancer at some point. So that sunburn that I mentioned, that actually causes 10 permanent mutations per cell per day that you get sunburned. Wear your sunscreen. I don't want to risk having 10 permanent mutations per cell just to go out to the beach. Sunscreen is relatively inexpensive. Treating skin cancer is not. Yeah, I know, it means I'm pale all the time, but I'm going to be anyway. I'm genetically incapable of tanning. So the sunscreen is worth it. So I told you that we looked at mutation rates and they vary by organism. That's actually what this is getting at. So we're actually looking at various different kinds of organisms in these graphs. So you've got a bunch of different colored dots. You've got the eubacteria, which is this kind of really dark cluster down here. You've got, let's see, the rest of them, the archaea, that's this sort of green dot. You've only got kind of one dot there for those. You've got a bunch of viruses, which are over in this region. So the viruses, you can tell, they're higher up on the graph. They have a higher mutation rate than the bacteria. That's because they have no correction mechanisms. So bacteria actually have a lower mutation rate than viruses. The next graph, as we look over on the right-hand side, you'll notice that we've got, let's see, bacteria, archaea. They have low mutation rates. And then when you look at mammals, like us, we have a higher mutation rate than bacteria? What gives? We have bigger genomes. We have a lot more DNA. So the sheer absolute numbers of mutations that we can accumulate is higher because our genomes are bigger. So they didn't put mammals over here because they couldn't necessarily fit everything on the same scale, and that's actually why we've got two separate graphs. Please note, you've got two different number sets on this y-axis. This one ranges from 10 to the minus 2 all the way up to 10 to the 7th. This one goes from 10 to the minus 1 to 10 to the 2. Pay attention to your scales. Pay attention to your axes. So overall, what you see is this dot here for archaea and that dot there for archaea, and they're both actually pretty close to 10 to the minus 1-ish. They're at about the same place. The mammals, we are so far different than some of the other stuff that it would have been very hard to get that on the same graph. So again, we're actually looking at the numbers of mutations per genome size. Because we have bigger genomes, we have more mutations. Viruses have smaller genomes, but no spell check mechanisms, so they have the most relative mutations. I also told you that it varies by gene and by tissue, even within our bodies, and that's actually what this table is getting at. It's table 6.1 from your textbook. So what they're looking at is the mutation rate per letter of DNA in different tissues. So, for example, if you look at Homo sapiens and you compare them to mice, if you look at the germline, that's the reproductive cells, eggs and sperm, you end up with about 216 mutations for humans and about 39 for mice. All right. Let's see. Germline mutations for C. elegans, flatworms, 9. They have smaller genomes. That's okay. But if you look at like what happens in our intestinal epithelium versus our retina, you can clearly see that we get way more mutations in our intestines than we do in our retinas. Guess why? Really critical, guts are slightly less critical. Mutations, changes in our retinas usually lead to blindness. Mutations in our intestines don't have quite the same kind of impact on our survival. So they actually estimated this in mutations per generation. And so, for example, every generation, the genes for our retinas are estimated to have about 54 new mutations, or almost 1 per cell division. But if you look at our intestinal epithelium, that is 160 per generation, but only 0.27 per cell division. Our intestinal cells also grow faster. So all of this kind of plays into it. Most of the time, we get to observing mutations, we find out about them because they make a gene quit functioning properly. So we end up with a disease like cancer, or like blindness, or like sickle cell anemia. So we usually observe loss of function. And this doesn't include the silent mutations, the replacement mutations, the synonymous mutations where the amino acids don't change. No change in amino acid means no change in protein, means no change in function, and natural selection doesn't care. So the other part of this is that some replacement mutations, even if you swap out an amino acid, it may have little impact, no impact, a lot of impact. It's kind of hard to tell. They may weaken or even possibly strengthen the function of that particular protein. So, for example, in C. elegans, in both nuclear and mitochondrial genes, they found an average of 2.1 permanent mutations per the whole genome, in the whole genome of these flatworms per generation. In our mitochondria, or even in the mitochondrial genes of the flatworms, they found a 10 times higher mutation rate. And that's because mitochondria have their own DNA, but it doesn't have the same spell checking mechanisms that our nuclear DNA does. So they accumulate more mutations. They have less repair machinery. Okay, last but not least, this actually looks at whether mutations are good or bad, and it also looks at whether where mutations tend to kind of be. So looking at part A of our figure here, what you're actually looking at are genes on a bacteriophage, a virus genome. And you can see that there are these kind of clusters, these areas where you have a lot of mutations, and then these areas where you have relatively few mutations. So even within a chromosome, you have hot spots for mutations or genes that are more prone to mutating than others. Again, it's generally thought that these are kind of less critical genes. They're ones that aren't necessarily as related to our fitness. When we actually kind of plot this out, which is what we're looking at in the bar graph, what you have is a fitness of 1, which means it has no impact on survival or reproductive success. Now, look at what is below 1. These mutations are bad. You have some mutations that are slightly bad, some mutations that are pretty bad, and some mutations that will flat out kill you. Lethal mutations. They do occur. You probably don't know about it because you wouldn't even be born. In contrast, there are also some good mutations. There are a few mutations that are beneficial, that actually, I don't know, confer flu resistance or cold resistance. Most of our mutations, about 70% of the ones that we've observed, actually reduce our fitness, but only by a little bit. It's typically about a 2% reduction, which puts us somewhere in that range. 70% of our mutations occur in the neutral to no real major loss in fitness. A 2% loss in fitness means that instead of having 100 offspring, you're only going to have 98. That's just not a huge issue, no matter how you look at it. There are those mutations, and it looks like this is about 20% of them, that are pretty darn lethal. Again, those are major, those tend to not be survivable. So this is where we leave off with the basic review of genetics and DNA biology. The next chapter is going to help us connect this information back to mechanisms of natural selection and looking at how much of an influence this actually has on the genetics of whole groups instead of just like looking at us passing our characteristics on to our offspring.